thank you very much for the kind introduction. And I'd just like to start by congratulating the organisers on organising such a wonderful meeting. Some of the talks have been truly excellent and really cutting edge, and it's been very interesting so far. And secondly, I'd like to thank Shiseido for inviting me to speak at this luncheon seminar, and I hope you'll enjoy your lunch while I'm talking about some of the work that I did during my postdoc and some of the work which we're doing in my lab at Imperial. So this is what we're interested in, human scalp skin and specifically human hair. Just to orientate everybody, on the top we have the epidermis, which is composed of keratinocytes. This is an ectodermally derived structure. Beneath this there is the skin dermis, and then the dermal white adipose tissue, otherwise known as the fat. And within this you can see that the skin is full of hair follicles, specifically as this is taken from the scalp. Now, even though the hair follicles are comprised of many, many different cell types, they're just comprised of two tissue types, epithelial tissue and mesenchymal tissue. So the epithelial compartment contains the outer sheath and the bulb shells, as we all know very well. And then within the mesenchyme of the hair follicle, there's the dermal papilla, the dermal cup, as we heard about yesterday, and also the dermal sheath, which surrounds the outside of the follicle. So this is adult scalp skin, and as you can see, it's full of hair follicles. But all these hair follicles are actually formed prior to birth as a result of interactions between these two tissue types, the mesenchyme and the epithelium, in a process known as secondary induction, which enables formation of follicles. So you start off with a very simple epithelial and mesenchymal tissues adjacent to one another, and as a result of signaling from the dermis to the overlying epidermis, we see induction and development of hair follicles and also other ectodermal appendages. So as I say, we start off with this simple structure with epithelium and mesenchyme, and within the mesenchyme there are fibroblasts, and a broad signal, potentially a Wnt signal, is believed to signal to the overlying epithelial cells, causing localized thickenings known as placodes, which form where the hair follicles will go on to appear. And after this initial signal from the mesenchyme to the overlying epithelium, the placodes send a reciprocal signal down into the mesenchyme and result in migration of these fibroblasts together towards the signal so that they double in density and form a dermal condensate. And then later on in development, in response to a signal from the condensate to the overlying placode cells, they proliferate and they start to push down into the skin, firstly forming the hair germ and the hair peg, and later the hair follicle. And you can see as this process is occurring, these condensate cells become engulfed within the follicle and now become known as the dermal papilla. And at the same time as this encapsulation of the dermal papilla cells is occurring, these interfollicular fibroblasts, which do not incorporate into the follicle, also begin to diverge, and we start to see appearance of an upper layer of fibroblasts and also a lower layer of dermal fibroblasts within the interfollicular dermis. And so these interactions between the mesenchyme and the epithelium are really important for hair follicle development, but they're also really important for hair follicle cycling and transition through the stages. So we have anagen, which is the active growth phase, which enables growth of long hair on sites such as the scalp, and shorter hair on other body sites such as the eyebrows or the eyelashes. Catagen is the regression phase, and then in telogen, the resting stage of the hair cycle. The dermal papilla, the mesenchyme, and the overlying epithelium, specifically the bulge and the hair germ, are in very close proximity to one another. And for the follicle to exit telogen and re-enter the anagen phase, you again need interaction between these two tissue types within the follicle. And it's believed that a signal from the mesenchyme activates the overlying epithelial cells, specifically the hair germ, causing them to proliferate, and they push down, and the follicle re-enters anagen. So you can see that there's a lot of commonalities between hair follicle development, where we get interaction between the placoid and the condensate, and hair follicle cycling, where we get interaction between the dermal papilla and the hair germ. And the dermal papilla is really important for this transition. It needs to be physically present for the transition from telogen to anagen to occur. So Valentine and Greco showed very elegantly using mouse models that this was the case. And she took mouse skin in telogen, the resting state. The green cells are the epithelium, the bulge, and the hair germ. And the red cells are the mesenchyme, the dermal papilla. And she used lasers to ablate, specifically ablate the dermal papilla in certain follicles, but not others. And you can see that if this is over time, then you can see that if the dermal papilla remains, then these interactions between the compartments occur, and the follicle can re-enter anagen after a period of time. However, if you ablate the dermal papilla, as in these two examples here, then no interactions between the compartments occur, and subsequently these follicles get stuck in telogen. And the dermal papilla is really the key compartment here, because she also repeated these experiments, but instead of ablating the papilla, she ablated either the bulge or the hair germ. And when she ablated either of these two epithelial compartments, compensation from the other epithelial cells within the follicle enabled the follicle to regrow and regeneration to occur, and transition from telogen to anagen to still proceeding as normal. So we do also see a kind of ablation in the human hair follicle in the form of androgenetic alopecia, also known as male pattern baldness. And here, over time, we see this miniaturization of the follicle, resulting in much smaller hair fibers exiting the skin surface, but underneath the surface, there is also a shrinkage of the dermal papilla. And this is due in part to a loss of extracellular matrix from the dermal papilla structure, and also a loss of cells from the dermal papilla. And it seems very unlikely that these cells are lost due to apoptosis, and it's more likely that they're lost due to migration out of the papilla at maybe during anagen 6, or maybe during another part of the hair cycle, as Ralph suggested yesterday. So why is it interesting, or why should we study this? Why do we want to work out what the role of the dermal papilla is in hair follicle development? And so, well, many people have androgenetic alopecia or male pattern baldness, and whilst hair transplantation is suitable therapy, you do need the donor area to have enough follicles to transplant into the recipient balding area. And sometimes this is not the case, so not all candidates are suitable for hair transplantation from the occipital to the frontal scalp. And we also want to know how to regenerate hair follicles, because we're interested in establishing follicles in both skin grafts and scars, which are devoid of hair follicles, as we believe this will be the first step to making scars and skin grafts, which are essentially biological bandages, more functional. And we believe that the presence of a hair follicle within a scar or a skin graft will in turn promote vascularization and innervation of these tissues, as I said, and making a functional skin. So what are the different strategies we can use to try to make new hair grow, particularly on the frontal balding scalp? So we want to either biologically regenerate tissue with new or renewed formation, and there's two particular cell therapies which people are investigating to try to achieve this. One is based on the idea that you can inject cells with inductive potential that will act a bit like a dermal condensate, and in turn promote entirely new formation of a de novo hair follicle. And this is termed induction. Alternatively, if there are still miniaturized hairs on the frontal scalp and you inject inductive cells, there is this idea that these inductive cells can incorporate into miniaturized follicles and enable them to increase in size thus regenerating their original form and thus function. And in my talk today, I'm going to focus on our work to look at inductive capacity of hair follicle dermal papilla cells, and then Dr. Kishimoto is going to focus on rejuvenation using dermal sheath and dermal cup cells. So if we want to study dermal papillae or other fibroblasts within the skin dermis, we first have to isolate them. And so we get tissue as discarded tissue after hair transplant surgery and other surgical procedures. And we isolate, we chop off the end bulb, as you can see here, and then we use this micro-dissection approach to invert the dermal papillae and expose the structure. And we can place this down into culture, and the intact structure will collapse, and the cells can migrate out, and then thus we can establish a culture of dermal papilla cells for use. Now, this procedure of obtaining hairs from the occipital scalp and moving them to the frontal scalp in hair transplantation can be done via two methods. You can either use a FUV punch graft to remove hair follicles from the occipital scalp and you don't get any scar, or alternatively you can use a strip method for isolating these cells and chop the follicles up into pieces, and then that does result in a scar on the back of the head. And so I just have a quick plug for a student in my groups, Magdalena Plotchek, is talking this afternoon in the concurrent session 13 on scar formation after excision of follicles from the occipital scalp, and what we can do about on how we can remodel these scars. So once we have our end bulbs cut off and our dermal papilla cultures established, we also need fibroblasts to compare as a control cell type. And so we like to also use a micro-dissection method to isolate fibroblasts, so we're consistent with our approach. And we chop off the fat, and then we transect the dermis into the upper and lower dermal layers and establish cultures from the explants of these. And when you passage these cells, you get either pretty indistinguishable cultures of either dermal papillary fibroblasts from the upper dermis or reticular fibroblasts from the lower dermis. Now, we're interested in intact dermal papillae and sheath because many years ago, Colin Yehoda showed that you could take, specifically in this example, dermal sheath and implant them into non-hairy tissue, and the intact dermal sheath was capable of promoting a full neogenesis or formation of an entirely new hair follicle. And this is some of our work looking at the inductive potential of dermal papilla cells, also intact. This is an amputated rat whisker follicle. It was amputated along this line here to inactivate it, and we placed against this 3 intact human dermal papillae, which you can see based on the staining for human nuclei specific expressing cells. So these 3 structures here are the human dermal papillae, and you can see that only the one that is directly adjacent to and touching the inactivated epithelium is capable of interacting with it and thus inducing formation of, in this case, a new hair fiber. And the ones that reside down here just sit there, and there's no interaction with the epithelium, and we don't see 3 hairs forming. We just see 1. So intact human dermal papillae are inductive, as are dermal sheath, but I said that we also study these cells in culture, and they look as though they are relatively indistinguishable from one another. However, we've been trying to develop a set of markers which we can use to distinguish between the cell types, and so we've stained with things such as podoplanin and CD26 and alpha-smooth muscle actin, and a range of other markers. And actually, the eagle-eyed of you will see that looking at just expression, we see relatively few differences between the dermal papilla fibroblasts at the top and the reticular fibroblasts in the bottom row here. But we found, when we looked at a specific marker, nucleolin, which marks regions of transcriptional activity, within each nuclei you get spots of nucleolin, And dermal papilla cells contain on average 2 to 3 nucleopen spots, whereas both types of fibroblasts contain on average 5 nucleopen spots. And so combining expression profiles together with nucleopen profiles, we can accurately distinguish between these different cell types which we're analysing. And a few years back, Colin Yehoda also showed that dermal papilla cells can turn into bone in culture, but he never looked at other fibroblast subtypes. And so we've also been looking at other ways to distinguish between these fibroblast subtypes. And we found that when we grow dermal papilla cells, and papillary or reticular fibroblasts in growth media, nothing happens. But when we grow dermal papilla cells in osteogen media, they very quickly turn into bone, much faster than mesenchymal stem cells, as Colin had shown. But we don't see any bone formation from the reticular or the papillary fibroblasts. So this is something that is specific to the papilla. And despite there being these clear differences between cells, one thing that all of these fibroblast types have in common is that in culture, they lose their capacity to induce new hair growth. So despite intact papillae being productive, cells in culture very quickly lose their ability to make new hair grow in recipient epithelium. And this is just profiling that we did, which really highlights the transition that these cells go through when you put them in culture. Every line on here represents a gene which was perturbed in its expression when we took dermal papillae and placed them in culture. All the red lines are genes which were expressed at very high levels in the intact structure and decreased in culture and then stayed low throughout passaging. And then blue lines represent transcripts which were expressed at low levels in intact papillae, which increased in culture and stayed at high levels throughout passaging. So even though our cultured dermal papillae cells lose the ability to induce new hair growth, there is some clinical work which suggests they still do maintain an ability to rejuvenate miniaturized hair follicles, which will be focused on later. So when I was a postdoc in Angelo Cristiano's lab, I found that the way that you group cells could really influence whether or not they were inductive. And we found that if you group these cells on the underside of a petri dish in 10 microliter drops with 3,000 cells per drop, and then you flip the dish over, these cells would aggregate into a dermal spheroid. And we found that this, in turn, partially restored the intact signature of dermal papilla cells. So again, this is this transcriptional plot. So all the genes which either increase or decrease in expression in culture, 22% of these genes are actually have their expression profiles restored when we grow dermal papilla cells in hanging drops to form spheroids. And we in turn went on and showed that this also partially restored the inductive capacity of these cells in culture. And so following on from this is what I did with Savon Harrell, also in Angela's lab. We used a patch assay to look at inductivity. It's just a different form of inductive assay where you combine your inductive cells that you're testing together with murine epithelial cells. And we know from Young Kanson's work that dermal papilla spheres are inductive in this assay. But when we grew dermal papilla spheres in either ruxolitinib or tofacitinib, which are JAK inhibitors, we found that with ruxolitinib we didn't see any effect, but when we grew the dermal spheres within tofacitinib for 48 hours and then put them into this inductive assay, we could significantly enhance the number of hair follicles that we were seeing being formed. And we performed transcriptional analysis on these tofacitinib and non-tofacitinib treated spheres, and we found that growth of the cells within tofacitinib makes the dermal papilla spheroids even more like the intact dermal papillae than just regular dermal papilla spheroids in terms of their transcriptional profiles. Now, growth within a spheroid is not real and end-all. It doesn't make other cell types inductive. So despite us knowing that papillary fibroblasts and dermal papillae come from the same upper lineage progenitor, if you grow papillary fibroblasts in these hanging drops to get formation of dermal spheroids, they form spheroids at about the same rate as dermal papilla cells do. However, this does not enable them to become inductive. So spheroid formation is necessary for dermal papilla cells to be inductive, but not sufficient for fibroblasts to become inductive. And so we're asking now, why is this? And assessing expression alone will not tell us how everything about a cell. It will just tell us what is expressed or not. And so within every cell, within every nucleus, obviously you have DNA and it is packed within different conformations. And so you get these regions of closed chromatin, which are inaccessible to transcription factors, so they cannot bind and elicit expression. And then you get these regions of open chromatin, where enhancers and promoters are in an exposed state, and transcription factors can come in and bind and enable expression of specific genes of interest, and potentially those related with trichogenicity. And so we hypothesized that there are specific chromosomal rearrangements and opening of chromatin, which are induced by us growing cells in a 3D structure, but there will be specific ones for dermal papilla cells, not dermal fibroblasts, that are potentially related to the acquisition of inductive potential within these fibroblasts. And so, to try to answer and detect what these chromosomal rearrangements might be, we're performing two types of sequencing: RNA sequencing to assess transcriptional activity, and attack sequencing to identify regions of open chromatin. And we are comparing fibroblasts and dermal papilla cells in two-dimensional and three-dimensional cultures. So the fibroblasts are not inductive in any state. The dermal papilla cells are not inductive in 2D, however the dermal papilla cells are inductive in 3D. And we're hoping to pass out the effect of 3D culture with the contribution of specific cell types to inductivity, to uncover how 3D culture conditions can confer inductive potential to these cells. And so we spent the last year trying to optimize library preparation for attack-seq. It's actually quite difficult. With RNA-seq, it's easy. You just extract RNA, and if you have good quality RNA, this has a RIN score of about 9.9, I think, we can send it off to a company who will perform RNA-seq for us. With attack-seq, we've decided to create the libraries ourselves, and now we can remove primer dimers using SPRI beads to remove small and large fragments from our libraries. And we get nice nucleosome banding, and all of our samples have now been sequenced, and we have all the data, and we're inputting it through both RNA-seq and attack-seq pipelines to try and identify a chromatin which is specifically opened in three-dimensional cultures in dermal spheres. So why are we doing this? Well, with regard to cell therapy, I've learned over the past year or so, I was quite naive about this in the beginning, but I've realized that any change or modification you do to a cell significantly increases the regulatory approvals you need to get to use it. So using cells in a two-dimensional state for cell therapy is relatively easy. Using cells in a three-dimensional culture makes it a lot more expensive. And so if we can work out what it is about 3D culture which promotes inductivity, we can potentially re-engineer cells in two-dimensional culture to think they're in a three-dimensional culture and make them more inductive. And relating to this, I think we have to think about what type of hair we actually want to induce. If we just try to induce new growth of hairs on the frontal scalp, they will be again susceptible to DHT, and in time they will miniaturize. And hair transplantation works because you're transplanting, taking follicles from the occipital scalp and moving them to the frontal scalp, and this concept of donor dominance means that they still think they're in the occipital scalp and they never become sensitized to the DHT which is present in the frontal scalp. And so we specifically want to try to induce growth of occipital follicles in a frontal location so that they do not, over time, become miniaturized. And just looking at this, there has been work from Young Kwan Sun's lab in Korea, and he presented this earlier, that shows that occipital follicles are far more inductive when you grow them in spheres compared to frontal follicles, and actually beard follicles are the most inductive. We also heard from Ting Chen earlier that HOX codes potentially can control identity on different sites, and so potentially this is related to HOX codes. And in this paper, they specifically looked at SFRP2 and showed that there was a positive correlation with the inductive potential of dermal papilla cells and the inductivity and the expression of SFRP2. And related to this, Elaine Chu, A-Star, did some nice work profiling dermal papilla cells from balding and occipital scalp in culture, identifying numerous transcripts that were differentially regulated between the two sites, and we still need to look at how this relates to their inductivity. So I hope I've convinced you that dermal papilla cells are the key drivers in hair development and cycling, and loss of them results in miniaturization of the follicle and hair growth. Intact papillae are inductive, however they lose their capacity to produce hair when you grow them in culture, essentially making them indistinguishable from dermal fibroblasts. But when you grow cultured cells in a hanging drop to make dermal papilla spheroids, this enables them to regain their inductive potential, but it doesn't do anything to the fibroblasts in culture. So for cell therapy, growing cells in a two-dimensional culture will be much easier for implementation compared to growing cells in spheroids. And for long-standing inductivity, we should focus on regenerating occipital follicles on a frontal site. And then lastly, I'd just like to say thank you to my lab, and the work on ATAC-seq and RNA-seq, which we're doing now, is being conducted by Nick, and it is funded by the British Skin Foundation. And with that, I don't know if I have time for any questions, but thank you very much.
Now time to the question and answer. And maybe we can take one or two questions. Yes, please.
Thanks, Claire. In the Omics analysis, what do you think would be the influence of the epithelium in the genomic expression profile of your EP spheres? Because when they go into the skin, they actually then make a hair follicle, and presumably there's some crosstalk. So I just wondered whether it's worth also considering looking at the gene expression in the presence of factors that are coming from the epithelium.
Yeah, absolutely. And some groups have, one of the ways people try to make dermal papilla cells more inductive is to grow them with kind of epithelial factors. Yeah, we only saw 22% of genes which were restored in our spheres, but I'm sure if we so actually we're growing double spheroids now, and we put epithelium around the outside. And if we haven't done profiling yet, but I would presume that the dermal papilla cells will further become more like intact dermal papillae. It's just like development. You have the condensation, but you need signals from the epithelium to promote formation of the papillae and engulfment of the papillae by the epithelium. So the crosstalk is really important.
Hi Claire, what a wonderful lecture for a clinician, as I am. The question is, will the frontal environment induce a change of the size of this dermal papilla coming from the occipital skin? So are you sure there will not have any influence from the frontal side?
So it seems with hair transplantation, that's why hair transplantation works, because the environment does not affect the follicles once you move them to a frontal site. So the frontal dermis and the occipital dermis are derived from different developmental origins, and it seems when you take the follicle from the occipital and move it to the front, it just remembers where it's come from. So even though the environment is different, the follicles do not become susceptible to miniaturization. But some surgeons may want to comment more on this. But it seems that they, this donor dominance concept means that they are not influenced by the environment. Even if we don't know about that, but I feel that making an occipital follicle would be a better starting point. If you try to make a frontal follicle, they're not very, frontal dermal papillae are not very inductive in the first place. So it might be difficult to do one with frontal dermal papillae cells.
Do you think there could be any risks attached to this type of or the ultimate aim of this type of activity, implanting cells which can induce epithelial cell growth, bone formation, for example?
No. Nature knows best. Like if we manipulate them too much, sure, but hair development occurs perfectly normally during embryogenesis, and if we can recreate that process, then no. I think it would be good. If we manipulate them too much and try to express things which are not naturally in the dermal papillae, sure, then potentially you are going to induce other structures. And the bone formation thing is really interesting because the intact papillae is full of inhibitors of bone formation, but when you put them in culture, they suddenly lose all their inhibitors of bone formation and then they turn into bone. So the intact papillae, I think, would regain these inhibitors and potentially would not form bone formation. But yeah, so long as we can kind of engineer the system to recreate natural interactions, then I think it should be safe.
Thank you very much. Dr. Higgins?